As early as the 1920s, scientists began to recognize the importance of genetic diversity to the health of biological populations. Preservation of genetic diversity maximizes flexibility, allowing populations to adapt to many kinds of environmental change. The more alleles a population has, the better its chance of survival under environmental stress. However, it was not until the 1950s, when scientists discovered much more about the structure of DNA, that molecular genetics could be effectively used in research. While there are many ways of measuring biological fitness, looking at the DNA unlocks information which cannot otherwise be observed. This information, in turn, equips us to implement more effective and intentional conservation efforts. Many factors influence genetic diversity, but a lot more research still needs to be done to better understand how and why gene pools change.
Hi, I'm Elizabeth, and I'm a science research intern here at the Chicago Botanic Gardens. I'm using genetics to look at the effect of pollinators on gene flow through different plant populations.
For my research, I'm using two rare plant species of the genus Clarkia. Both species are endemic to California, and different populations have different primary pollinators. These include noctuid moths, hawk moths, bees, flies, and even hummingbirds. One factor that differentiates these pollinators is their flight range. A pollinator with a larger flight range is more likely to visit multiple populations which have different genetic material, thus introducing new DNA to each site. For example, hawk moths travel a greater distance than bees or flies. We hypothesize then that populations primarily pollinated by hawk moths would show higher levels of genetic diversity and lower levels of inbreeding. This would mean that some pollinators are more beneficial to the fitness of a population. To determine the genetic diversity of a population, I begin by extracting DNA from multiple plants in different populations. This process uses a series of chemical and thermal means to break down the cell, get rid of unwanted material, and precipitate the DNA. Next, I use an amplification process called polymerase chain reaction, or PCR. The enzymes involved in PCR act like a molecular copying machine, producing hundreds of thousands to millions of copies of a certain genetic sequence. Having more DNA from a specific region makes analysis much more reliable. A DNA sequencer then analyzes this DNA and gives me specific data. If our results show a correlation, it's important to remember that it's only a correlation at this point, not necessarily a cause and effect relationship. Further genetic research will tell us with more certainty if these two trends are related.
Molecular genetics offers an increasingly beneficial perspective on gene flow and population fitness. To learn more about all the amazing research being done here at the Chicago Botanic Garden, check out the link in the description.